For a biologist, a cell is the building brick of life, containing a number of different organelles, each accomplishing its specific tasks and together allowing the cell to perform its vital functions. From an electrical perspective, however, the internal structure is not very important, and the cell can be viewed as a conductive cytoplasm enclosed by an insulating membrane and surrounded by a conductive exterior. A small imbalance in ionic concentrations, with the surplus ions accumulating close to the membrane, results in the resting membrane voltage. Placing a cell into an electric field causes a local charge redistribution on both sides of the membrane. The result is the induced membrane voltage, which superimposes to the resting voltage and disappears after the field is turned off. With simple cell shapes, the induced membrane voltage can be calculated analytically. For a spherical cell, it is given by the Schwann's equation, which states that the voltage is proportional to the field strength and cell size and follows the cosine function along the membrane. For more complicated cell shapes, the induced voltage can deviate considerably from the cosine function and must be determined either numerically, using a powerful computer, or experimentally, using a potentiometric dye. One of the potentiometric dyes widely used for this purpose is Di-8-ANEPPS, a fast dye with excitation and emission spectra dependent on the membrane voltage. Di-8-ANEPPS becomes strongly fluorescent when bound to the membrane, with the change of the fluorescence intensity proportional to the change of the membrane voltage. This video shows the protocol for measuring the induced membrane voltage by using Di-8-ANEPPS. In this example, Chinese hamster ovary cells are used. Plate the cells in chambers at 100,000 cells per chamber in HAMF12 culture medium. Supplement